A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Explain stuff.
All right, so if you recall, the main idea of this lab was this enzyme called peroxidase. Peroxidase is the enzyme we're dealing with. And remember, enzymes are usually proteins, and peroxidase is a protein enzyme. And remember, enzymes catalyze reactions, right? They speed up a reaction. And in this case, the reaction it speeds up is the breakdown of the substrate, hydrogen peroxide. Hydrogen peroxide to, actually, 4 hydrogen peroxides become 8 water. So water is the product. Hydrogen peroxide is the substrate. Now remember, peroxidase works at a constant rate to break down hydrogen peroxide into water. That means it'll break one down, then it'll break another one down, it'll break another one down, but it breaks them down at a constant rate. Okay? Remember that hydrogen peroxide is clear in color, and peroxidase enzyme is clear in color, and water is clear in color. So everything is clear. So we need a way of monitoring the reaction. That's why we added something that changes colors. It's called gayacall. Gayacall is a color indicator. A color indicator. That means something that changes color to show a reaction or during the reaction. Now, gayacall is reduced at the beginning. It's a reduced molecule. Remember, Leo says Ger. Reduced means gains electrons. It has electrons, so it's colorless. Colorless. However, when peroxidase breaks down hydrogen peroxide, that releases this oxygen. That allows gayacall to become oxidized. Gayacall becomes tetragayacall. That's right. These four gayacall come together as kind of a square. Okay, they come together as 4. Tetragayacall. This is the oxidized form. And this is brown. Brown color. Okay? So we added gayacall as a color indicator. Why? Because during this reaction, peroxidase catalyzes the breakdown of hydrogen peroxide to water. That in turn causes the oxidation of gayacall, a reduced molecule, to tetragayacall, an oxidized molecule, which happens to also have color to it, and that color is brown. Okay. By the way, where was our peroxidase enzyme from? Remember, it was found in the turnip extract. Okay, that's where we got the peroxidase enzyme from, turnips. So this is the overall reaction that we are trying to monitor. Remember I said peroxidase works at a constant rate? That means how fast is it breaking down the hydrogen peroxide into water? How fast is it oxidizing the gayacall to tetragayacall? That is what we're trying to figure out in this lab. That is what we mean by the enzyme's reaction rate. The enzyme's reaction rate. How fast is peroxidase working to convert the reactants to products? Okay, if it's doing it quickly, then the clear reactants become brown color products fast, right? If the enzyme is not working, the reactants remain clear, right? Is there any reason for the products to form if the enzyme is not working? No. So you won't have any brown color. If the enzyme is not working, everything stays clear. Okay? If the enzyme's working fast, brown color appears fast. If it's working slower, brown color appears slower. If it's not working at all, there will be no brown color at all. Okay, that is what they mean by the enzyme reaction rate. And what does that depend on? That depends on this enzyme's folding. Remember, proteins have to fold into the correct three-dimensional structure. Do you remember that's called the tertiary structure? If enzymes are not folded into the correct tertiary structure, they're not going to function. Okay, so what do you think happens when we change things like temperature, pH, salinity, things like that? Well, let's take pH. We changed pH, right? What do you think happens at pH 3, pH 5, pH 7, pH 9? Well, that's what we were addressing in this lab. What happens at pH 3? Does the enzyme stay folded nicely and continue doing its job, or does it actually unfold? Does it unfold? Remember the term for that? The term for that is called denaturation. Denaturation. Denature means when the protein unfolds, right? And do you guys remember what I said in class? Once a protein unfolds, does it refold? No. So a denatured protein does not renature. And you need a protein to be folded correctly to function correctly. So peroxidase is not going to function correctly because it is unfolded. So wherever you see low rate, low enzyme activity, I don't know, pH 3, pH 9, remember you saw some low enzyme activity there? Well, that might suggest that the protein is denatured. That might suggest that the protein's not going to function correctly. And wherever you saw the highest activity, that suggests that that is the best for protein folding. That is the best for peroxidase activity.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.